this is a short little screencast to address a question that came up on Monday about one of the questions on the homework assignments, the Proctorio thing. It was the question about the number of exons, and this is a screen capture of the question in McGraw-Hill, and it's basically giving you a DNA sequence and then giving you an RNA sequence, and then it's asking you to identify the number of exons that are in this DNA, that are in the RNA sequence. Sorry, the number of exons that are in the DNA sequence based on the RNA sequence that is done. And so what I've done is I've just copied these down here in a Word file, and I've used Courier to basically show us what the sequences are. And we use Courier because Courier is consistently spaced, and so it's easy to line things up. So here is the 5' to 3' DNA and the 3' to 5' DNA. And so I'm just going to create some space between these so that we can fit our RNA molecule in here. And remember that when RNA is going to be being transcribed, the polymerase moves from the 3' to 5' along the DNA so that it can transcribe the messenger RNA in the 5' to 3' direction. So I'm just going to copy this and I'm going to put it right under here so that we have the template strand of DNA, and I'm going to code the template strand of DNA in green, and I'm going to put the RNA strand in red. Keep in mind that this is not a full RNA molecule. That's what these dots are here at the end. This thing is going to go on. But what we can do is we can get rid of the 5' guanosine cap, and then we can line up the RNA sequence underneath this. And we see that there's complementary base pairing. Thymine to adenine, cytosine to guanine, guanine to cytosine. This matches, that matches, that matches. But this thymine does not complementary base pair with this cytosine. So that means that some of the DNA was excised, was spliced out of the messenger RNA molecule when it went through post-transcriptional processing. So we can just start spacing down until we get to a point where this lines up with the complementary bases. And so we see cytosines matching up to guanines here. Then we have adenine, uracil. Uracil, that matches. Adenine matches here, uracil matches here, guanine matches here. But then we get to this point right here where guanine does not complementary base pair with adenine. So we need to move down until we get to a point where we have 2 thymines in the DNA. But those 2 thymines are right next to a uracil, but uracil doesn't complementary base pair there. So we need to go down once again until we find 2 thymines in the DNA. Now we see thymine, thymine, adenine, complementary base pairs with adenine, adenine, uracil, and then we have 3 thymines and 3 adenines. And so what this indicates is that the messenger RNA, the pre-messenger RNA that was coded for by this section of the DNA, which I will now put in orange, that messenger RNA has been spliced out. This messenger RNA here has also been spliced out. So we have an exon here, a section of messenger RNA that was spliced out, and I'll just put that as a highlight. Oh, the highlighting doesn't work. Anyway, a section right here that was spliced out, another exon, a section here that was spliced out, and then a final exon. And this is why the answer to the question was 3 exons, because we have 3 parts of the messenger RNA that remain after you have spliced it out. Hopefully that satisfies the answer to that question.